everyone and welcome to Wonk. Today we're going to be looking at microRNAs. All of the cells in our body contain the exact same DNA sequence. So why is a brain cell so different from a skin cell? This is because genes can be turned on and off at different points in time. MicroRNAs are short RNAs that don't make proteins but are thought to control which genes are turned on or off. Let's take a journey underground to look at how microRNAs affect gene expression. The processes for making proteins in the body are finely regulated by strict instructions. Different signals fire around inside cells and can create an environment that makes the cell know to produce more or less of something. This is much like following the tube map. Get on the wrong line and you'll end up in the wrong place. Each building block in our body starts as a piece of DNA, a double-stranded winding string of adenine, thymine, guanine and cytosine residues. This gets unzipped by a naughty enzyme called DNA helicase, unwinding the tangled DNA. This is further unwound by another enzyme called topoisomerase, and the two strands are held apart by proteins. Transcription is where a copy of DNA is made into an mRNA molecule by RNA polymerase. The mRNA sequence is then read 3 letters at a time in a process called translation, where the 3 letters code for one of the 21 amino acids. tRNA and a ribosome work together to put the correct amino acids together like a string to form a polypeptide. This polypeptide then folds and bends to make a protein before going off to its final stop. Gene expression pathways can be complicated, so scientists like to map them out. The London Underground has 11 tube lines covering 270 stations. Currently, there are 1,872 known microRNA genes in humans, which can produce over 2,500 different microRNAs. That is a big map. MicroRNAs are short interfering strings of information that can block the production of building blocks affecting gene expression. They're produced in the cell nucleus and are pushed into the cell body where they are free-floating. Think of them as sticky molecules looking for a friend, or a matching mRNA sequence. Once they find their friend, they will bind to the seed region. So how do microRNAs impact the journey? Think of the process of mRNA to protein as a journey on the underground. The trip to become Protein X starts on Central Line at Liverpool Street and ends at Epping. The underground seems complex at first, so it's easy to get on the wrong train or off at the wrong station. If you're lucky, you'll get help from a member of staff who, like a regulatory protein, will put you back on the right track. But if you don't find a regulatory protein and get off at the wrong stop, and get distracted by Greggs or Starbucks, you might bump into signals, bamboozling molecules, or even a microRNA. Before you know it, you're stuck with the microRNA, matching perfectly shared interests and shared seed regions. Once bound, this new pair of lovers can instigate a whole host of effects. But as with a bad Tinder match, things might not work out, and the gene may never be expressed at all. So that's how microRNAs can find a partner and affect how genes are expressed. I hope to see you next time for another topic explained. Remember to like, subscribe, and stay curious.